All right, so I told you on Wednesday when I handed back the exam that we had done a good job of getting ahead to account for days we were losing for Engage. And so today what we need to do is we need to finish our discussion of invertebrates, which is what chapter 28 of the text talks about. But the way I split it on Canvas is we had a module on protostomes and we had a module on deuterostomes, but they're only really one big group, one big phylum of invertebrates that are deuterostomes, and then there's a more minor group that is talked about very briefly in the textbook. So as we finish up our discussion of invertebrates, we have only two framing questions to deal with. Two framing questions to deal with today. First one, what are the hallmark deuterostome characteristics? So remember, deuterostomes and protostomes, they were separated long before genetic data were used to help hone in on animal phylogeny. And so they had already been separated for a long time, and the question is why? What are those characteristics that were used to group deuterostomes together? And then finally, how are echinoderms organized? And Echinodermata is our one phylum that is a big phylum of invertebrates that are also deuterostomes. Because the phylum that we are part of, most of the living species and most of the fossil species are vertebrates. There are some invertebrate representations in our phylum. All right. First question. What are the hallmark deuterostome characteristics? What are the hallmark deuterostome characteristics? First thing, you need to remember that deuterostomes have been separated from protostomes even before genetic data became available, right? This was one of the things supported by genetic data. Remember? What were the other parts of animal phylogeny supported by genetic data? So we had protostomes grouping apart from deuterostomes. What other groupings were supported? Yeah. Animals with radial symmetry apart from animals with bilateral symmetry. And then finally, sponges apart from all other animals. Okay? And so this is something you need to keep in mind. Everything else on this slide is going to pertain to the groups inside Deuterostomia. So when it says both groups, it is not referring to protostomes and deuterostomes. It is referring to both of the main invertebrate phyla within Deuterostomia, okay? So it may be a little bit confusing, and I'm sorry for that, but the both groups is not referring to protostomes and deuterostomes. We're already separate from there. This is all deuterostomes. So now within Deuterostomia, we can still further separate the groups based on development, okay? So within Deuterostomia, we can still separate them based on development, but first we'll go through the shared features of organisms within Deuterostomia. So both groups, and again, this is not referring to protostomes and deuterostomes, this is referring to both major groups of deuterostomes, echinoderms and chordates. Both groups have radial cleavage, okay? And so protostomes have what type, and this is at the 8-cell stage. When you have 8 cells that have all formed from the zygote, and you basically have 2 layers of 4 cells, protostomes have what type of cleavage? Spiral, and so the 2 layers are offset. Deuterostomes have radial cleavage. The 2 sets of cells sit right on top of one another. Both groups, again, not protostomes and deuterostomes, but both major groups of deuterostomes have indeterminate cleavage. Versus what type of cleavage do protostomes have? Determinate. And what that means is even at a very early point in development, the cell is already destined to become some type of cell. So even at the 8-cell stage, if you remove a cell from a protostome, development stops. At the 8-cell stage, if you remove a cell from a deuterostome, that cell you remove develops into an embryo, and the 7 cells remaining develop into an embryo. It's how you get identical twins. And then finally, both groups, again, not protostomes and deuterostomes, but both major groups of deuterostomes form the mouth from the second opening. That's what Deuterostomia means. Okay, this should be review. Yes? Cool. All right, so now we'll start talking about specifics of these phyla. Echinoderms. what you find is they go through a metamorphosis in their life history, and when they transition from larvae into adults, they lose their bilateral symmetry. So their larvae are very obviously bilaterally symmetrical. As they metamorph into adults, they lose that bilateral symmetry and take on a type of radial symmetry. Do you remember the type of symmetry on your exam that the answer was, we have not talked about this yet? What was it?
We haven't talked about it yet.
We haven't talked about it yet. You are absolutely correct. Here it is. It's called Penta radial symmetry. That's a weird color. Can you even see that?
Yeah.
Oh, no, yes, maybe. I don't know. Okay. So pentaradial symmetry. Radial symmetry on one surface, you can interact with your environment in 360 degrees. You have an infinite number of planes of symmetry. In pentaradial symmetry, you can still interact with your environment in 360 degrees, but you have 5 planes of symmetry. Wait, man, I can't draw this. There we go. And so you have 5 planes of symmetry. One here, one here, one here, one here, and one there. Okay, you have 5 planes of symmetry. Like a sea star, right, with 5 arms. Although even some sea stars have more than 5 arms, but they still have that pentaradial symmetry. Here's one group. It's a very minor group of Deuterostomes, although it still has these shared features as well. Hemichordates, they retain the bilateral symmetry and also retain the eucoelomate body plan. Yeah.
Are sea stars an example of pentaradial?
Yeah, they are. And they're an example of echinoderms. So echinoderms include, we'll get to this, this is our next framing question, but sea stars, sea cucumbers, urchins, sea lilies, and sea daisies. And then chordates. So this is the other major phylum of Deuterostomes, including humans, have bilateral symmetry and a eucoelomate body plan throughout their development, as well as some other features that are not shared with other Deuterostomes. Okay? A notochord, a dorsal hollow nerve cord, pharyngeal slits, a post-anal tail, and then there's another one that we'll talk about when we get into chordates next week. Yeah?
Are the pharyngeal slits like gills?
They can be. So pharyngeal slits can develop into gills, or they can develop into something else. So we don't have gills, but we do have pharyngeal slits. Early in development, we have slits that develop into some of the soft tissues and connective tissues inside of our trachea and pharynx. All right. So I have a question for you all to work on in a group. So take 2 to 3 minutes, and I want you to come up with a really good answer to this question. So now, knowing that we are Deuterostomes, and already we've talked about us being bilaterally symmetrical, and it's on the slide to remind you, and we are eucoelomates, again, on the slide to remind you, and we have 3 germ layers. Okay, so thinking about some of these features that we share with some animals and not with others, I want you to come up with a good way to answer this question. Why did we develop in similar ways to echinoderms, but differently than the other segmented phyla? So remember last time, and I'm pretty sure it was on Monday, we talked about 3 segmented phyla. What are they? Well, those are the 3 tagmata within Arthropoda, but the 3 segmented phyla. Arthropoda, Annelida, and Chordata. Okay, so our phylum, arthropods, insects, spiders, crustaceans, centipedes, millipedes, horseshoe crabs, vinegaroons, right, all those wonderful animals. And then annelids, earthworms, leeches, clamworms. And so why do we develop in similar ways to echinoderms, sea stars, and urchins, and develop differently than other segmented phyla? Okay, so I want you to take 2 or 3 minutes, talk with those around you, and I want you to come up with some potential solution to this question. All right, 2 to 3 minutes, starting now. Do you play soccer, Shelby?
What?
Do you play soccer?
I do.
Okay, I'm sorry.
That's okay. Don't worry. Similar to other things, so like we can't have the indeterminate cleavage formed by diagonal.
We usually do. Oh no? We usually? Yeah. We have that put on the skull. We get the skull right here. What? Oh, we're going with that.
You have an answer?
This is true. Well, you know it.
The unisolar seeds.
Got to show up to class early. But I like this, filling up the middle of the classroom. Yeah. Thank you.
We won't have physics left.
Oh, yeah, sorry. They were just saying stuff.
This is the only saving grace here.
This is the only thing we're still there.
Just about. Unless it
All right. How have we answered questions like this in the past? When we ask the question of, why are so many animals bilaterally symmetrical and use coelomate like we are? Yeah, Emerson.
We answered it in 2 ways, like
Okay.
God's design and natural methodology.
Right, so then you can answer it from methodological naturalism. The way to explain it only through natural processes is because we share a bilaterally symmetrical and a eucoelomate animal. Although that seems to be inconsistent with what Scripture teaches about origins. And so I think we need a better explanation. And so the other one is that is a biomechanical necessity. That if you're going to live an active lifestyle, you have to have a very efficient body plan, and you have to be able to move in a very directed way. If you're going to move in a directed way, you need to be bilaterally symmetrical. Okay? And if you're going to be efficient enough to have an active lifestyle, you have to be a eucoelomate. Okay? And you're like, what about nematodes? They don't live an active lifestyle, right? They just kind of hang out, and even the free living forms don't live a very active lifestyle. Okay? But we can't answer this one with biomechanical necessity. Right? I mean, do y'all know what urchins are? They don't live a lifestyle similar to most bilaterally symmetrical animals, and that's because they lose their bilateral symmetry, and yet they develop like we do. And so you can't answer the similarities in development based on biomechanical necessity. You have to answer it some other way. So what is that some other way? Yeah. I would love to repeat the question. Why would we develop in similar ways to other animals and differently from others? So why would we, in this case, I made it more specific. Why would we develop similar to echinoderms, right? To where, the second opening in the gut forms the mouth. Cleavage is determinate and radial when we are nothing like echinoderms. I mean, the last time I checked, Mika and then Micah.
Because we live where
Potentially, you couldn't do it for humans. You could do that more generally for chordates. The overwhelming majority of chordate species are marine organisms, and all of your echinoderm species are marine organisms. Okay, so you could do it for chordates generally. You could not do it for us specifically. Although there are other marine organisms that don't develop like we do. Micah.
Could it be related to Hox genes?
Maybe, right? These master control genes. And so if we had similar numbers of master control genes or similar types to echinoderms, that would certainly explain why we would develop in similar ways. But then you just kind of have pushed that question back a little bit, and then you have to ask, why do we have similar Hox genes to echinoderms? Now keep in mind here, it tells us that hemichordates, they retain their bilateral symmetry. Echinoderms lose their bilateral symmetry, but it's a very clear loss. Their larvae actually do live very active and efficient lifestyles as zooplankton in the water column, and then go through metamorphosis to take on a more simple body plan. So the larvae actually do have the same biomechanical requirements that we do. So I told you we couldn't answer this using biomechanical necessity, but that wasn't exactly accurate if you also include the larvae. Now, so then there's a question of why would these groups of organisms lose their bilateral symmetry? And we could talk about that when we talk about echinoderms, but there are still some of those issues, although we still have the problem that of all the bilaterally symmetrical eucoelomates, some of them are protostomes, some of them are deuterostomes, right? They all have to have efficient body plans, which is what being a eucoelomate requires, or requires you being a eucoelomate. And they all have to move in a very directed way, which requires you to be bilaterally symmetrical, and yet we still group them separately. They develop in different ways. It's interesting. The question is why? And what you find is that there's no real selective advantage to developing in any particular way. Just the first opening becoming the mouth or the second opening becoming the mouth doesn't make you any better equipped to face your environment. So it's a developmental difference. There's nothing super meaningful about it except that it separates the two groups. Here's what's cool. I will tell you this. First of all, a lot of the developmental differences that your textbook will point out between deuterostomes and protostomes are not true for vertebrates. These three are. Okay, these three also are true for vertebrates, including us, but a lot of the developmental differences between the two groups are not true of vertebrates. In many ways, vertebrates develop more like arthropods do. Okay, so that's one interesting thing, but let me tell you this. The overwhelming majority of our developmental research does not have to be done on humans or any vertebrates because there are other animals that are developmentally similar to us. The overwhelming majority of developmental research that we have used to understand human development has not been done on human development, but instead has been done on echinoderms or hemichordates or other invertebrate chordates that then we use as a model organism to explain development in humans. You do enough development in humans to understand that the development is similar to these groups, and then a lot of the research is done on these groups because you don't have as many ethical concerns and it's way less expensive. Is that a reason why? I don't know, but it is something that's very interesting to point out, that this gives us literally thousands of species that we can study development on that nobody has significant ethical concerns about. And yet their development is similar enough to human development that it's meaningful and gives us information that's helpful. Is that a reason why? I don't know, but it is convenient, right? It is convenient. So that's just something to keep in mind. All this to say, I don't know. I don't know a really good way to answer this question. except for maybe that it gave us an opportunity to have model organisms to study human development. Yeah, Micah. Will this be on the test? I mean, these questions are always potential for being on the test. Yeah, are you asking, like, if this appears on the test and I write I don't know, will that suffice as an answer to this question? I think that might be what you're asking. And if you say I don't know and give a reason why you don't know, I would be okay with that.
Okay, so that's how we answer this question.
Yeah. I mean, if all you wrote was I don't know, the directions say answer these questions in 3 to 5 sentences, and I don't know is 1 sentence. Unless you put a period after all of them, but then they're still not complete sentences. A period does not a sentence make, right? I know, blows your mind, right? Sentences need more than just periods. All right, any questions about this question? All right. So the next and the final framing question that we're going to deal with from chapter 28. Okay, and then we'll transition to chapter 29, not today, next week. So how are echinoderms organized? True or false, echinoderms are deuterostomes? True. Look at that. Some of you have learned something, right? Same day applying knowledge. I love it. Okay. Okay. So one big feature of echinoderms, other than adults having Penta radial symmetry, is a structure known as the water vascular system. Now this develops from the coelom, and what is that coelom again? What is it? It's a cavity. And it's a cavity in addition to the gut, right? And these are true coelomates, meaning what lines that cavity on both sides? Mesoderm. Meaning you can build a very sophisticated cavity with really sophisticated connective tissues and muscle tissues and all sorts of specialized tissues on either side of the cavity. In echinoderms, that cavity develops into the water vascular system. And so in this system, water moves and circulates from a central ring all the way down into radial canals, and then in some of these organisms down into tube feet. Have any of you ever been to an aquarium where a sea star was stuck to the glass? You see the little feet? Hundreds of tiny little feet on the bottom of the sea stars. Not the arms, right? But tiny feet on each arm, sometimes in the hundreds. And so those are basically one end of the water vascular system. The other end would be the central ring where the water enters. Yeah.
What about like octopi and stuff like that? Are those?
So those are Mallis. Yeah. So, what were you going to ask about a water vascular system?
No.
No.
For the,
Oh, okay.
Penta radial.
Yeah. So those are Mallis, display very obvious bilateral symmetry. All right. So inside of this water vascular system, it functions in locomotion, right? If you've ever watched the tube feet. If you haven't, find a video on YouTube of tube feet in a sea star. It's interesting just to see hundreds of little feet with suction cups on them propelling it across the glass. Propelling seems like a misnomer because they don't move very quickly. But, I mean, anyways. But it also works in gas exchange and nutrient exchange. So it functions as both a circulatory system and basically their equivalent of a muscular system. Okay? So it functions in locomotion, like is what our muscular system does, and it also functions to facilitate gas and nutrient exchange, which is what our circulatory system does. Now, abandoning the bilateral symmetry allows for a very simple body plan. And you'll see this when we dissect sea stars later in lab, is you'll see when you open this up, it has a very simple organization and not a lot of really complex tissues like you find in animals that keep their bilateral symmetry into adulthood. So when they give up bilateral symmetry, they lose cephalization. That's something we talked about before, having a defined head region that goes hand in hand with bilateral symmetry. So if you give up bilateral symmetry, you give up cephalization. So sea stars do not have a head. despite what Patrick would lead you to believe. Oh, Patrick. All right. Sorry. They also, losing that defined head region, you cannot really have a central processing center. Okay? Animals that have a central processing center have a defined head region. So if you lose the head, you lose the central processing center. So you lose the sophisticated responses that you can give to environmental stimuli. Now that does not mean that echinoderms cannot respond to stimuli, because that's one of our characteristics of living forms, right? If you are a living organism, you have to respond to stimuli. It just means their capabilities are limited in terms of what they can do. Just like a jelly. A jelly can respond to environmental stimuli, but those responses are limited. Have any of you watched a sea star pry open a mussel and evert its stomach inside the shell and digest the mussel? And then, oh gosh, it's awesome. If you ever watched a video of a sea star everting its stomach inside of a mussel shell and then the mussel slams its shell closed and rips the stomach off, that's a fun one to watch too. Anyways, sorry. Got to drop a couple of those things every class. All right. So echinoderms, this is a phylum, Echinodermata, and they are organized into 5 classes. And yes, it would be helpful to be able to describe the 5 classes of echinoderms, anticipating a question that you might ask. Just like in Mallis, we talked about specific classes, which we haven't tested on chapter 28 yet either, but it's helpful to be able to communicate about the various classes of Mallis. Right? What are our 3 main classes of Mallis? Those that are most speciose. Bivalves, gastropods, cephalopods. Okay? There are also polyplacophorans, monoplacophorans, but those are more minor, not as speciose. All right, so 5 echinoderm classes. First, asteroids. The sea stars. Yes, spelled the same, very different meaning. Asteroids have an evertable stomach and thick, what are called ambulacra, these really heavy sections on the bottom of the animal. And that's where the tube feet are, lining these ambulacra. Okay, sea stars. Ophiuroids. These are brittle stars. So in the middle of the animal, it looks like a sea star. And then it almost looks like a vertebral column radiating out from that central disc. It's not a vertebral column, but it looks like that, articulating calcified plates. These are pretty cool animals. Still very obvious pentaradial symmetry. Echinoids. These are urchins, hard urchins, sand dollars are also in there. I don't know if you knew this or not, but sand dollars are animals. And if you find them when they're still alive, you'll see hundreds, if not thousands, of tube feet on the bottom moving around, pulling sand into the digestive system of the animal. Echinoids, urchins, hard urchins, sand dollars, these lack arms and they have a really thick test. Again, all of these 3 form calcified plates, but in urchins, those calcified plates fuse together to form one big external skeleton. Echinoids sometimes have tube feet, but they don't move using tube feet, they move using big spines. Have you seen urchins? Yeah, anyways, if you haven't, you should look them up because they're pretty awesome. Crinoids, also known as feather stars, are filter feeders, and they still have that obvious pentaradial symmetry, and they have these long feather-like arms that radiate out from the mouth and trap material and bring it down. In some ways, they look like a modified anemone. And finally, holothuroids, cucumbers, lack arms, and they still have pentaradial symmetry, but they have what you would call a functional bilateral symmetry. They move in only one specific direction. Okay? Any questions about these 5 classes? Yeah, Cameron.
Does this like encompass all of stuff, like
All of echinoderms. All living echinoderms. There are 2 groups, there are 2 classes of extinct echinoderms, but these all living echinoderms are in one of these 5 classes.
So like for the asteroids, they don't have to just have 5.
Correct. They can have more than 5 arms, but they still have the pentaradial symmetry.
So that would include like the Crinoids.
Yes. Which can have like 20 or more arms, but it still has pentaradial symmetry. If you turn it over where the mouth is and you look closely, you'll still see that pentaradial symmetry. And actually, I believe that there are no limit to the number of arms that you can actually have in a crown of thorns sea star, because if you remove an arm, it'll usually grow 2 in its place.
Oh wow.
It's like the Hydra. I think the Hydra is if you removed 1, it grew 3 in its place, right? Or was it 2? 2? Okay. Sorry. So here's a representation of the water vascular system. Here's the opening to the water vascular system, the madrepori, and then here are all these radial canals radiating out into the individual arms. And here you have a tube foot with the suction cup. Again, you can have hundreds, if not thousands, of these on any given echinoderm. You can also see the digestive system also radiating out into the arms, maximizing the amount of space for digestion. And then here are your 5 groups of living echinoderms: asteroid, ophiuroid, echinoid, crinoid, holothuroid. Okay? Asteroidea, ophiuroidea, echinoidea, crinoidea, holothuroidea. Right? Wonderful names for these classes. They just roll off the tongue. Make so many connections to things you know already that you're just going to have no issues remembering these 3 groups. 5, not 3. Look at these 5 groups. Right? Okay? So keep in mind, these echinoderms, these are deuterostomes. We are deuterostomes. We share some developmental features for them. And I think the only satisfactory way that I can think of to explain why that is the case, other than that we share a more recent ancestor than we do with any other animals, which I don't think is a satisfactory answer, according to what I believe to be true, is that it gives us a number of species that we can use as model organisms to represent human development. Which is nice, because it makes developmental biology less expensive, less ethically concerning, and still really beneficial for understanding how human development happens at the earliest points of development. All right? Questions? Thoughts? Concerns? Okay, we have finished chapter 28. On Monday, we will begin chapter 29, and we will begin talking about vertebrates, and we will continue talking about chordata, which we mentioned a little bit already.